I'm going to talk today a little bit about differential transcript usage analysis and why and how we have been doing it so far, and in what ways our methods might be proving insufficient given the current and future directions of RNA-seq experiments, and in what ways we try to tackle some of these problems with our current new tool called SPIT. So to start with some definitions here, we have a standard gene going through transcription in these two cells. The polymerase attaches, the spliceosome forms, and then suddenly the second cell here decides to skip this third exon to produce a different transcript and consequently a different protein. So if you were able to quantify the relative contributions of these transcripts to the overall expression of this gene in these two cells, you would see that the dominant isoform for this gene changes between the two cells, and that is what we call a differential transcript usage. It's when the second cell here decided to use a different transcript for the same gene. And I'm going to throw in another term here, which will be useful for our future slides, and it's called an isoform fraction. And it is simply the ratio of the total gene expression that one isoform accounts for in a sample. So for example, in the second cell, you have about 100 units of this gene expressed, and about 75% of that is accounted for by the second isoform. So we're going to say that the second isoform here has an isoform fraction of 0.75 in the second cell. And what DTU is not, however, is a simple change in the expression values of individual transcripts. So if you were to look in this picture and analyze individual isoforms, you would see a significant decrease in their expression profiles in the second cell, but because their relative proportions to each other does not change, we do not consider this a case of DTU. And when we do this type of analysis, it is rarely between two individual cells. It is more common to compare groups of cells, and even more common to compare groups of individuals, where one group is usually the control and one group is the case, which belongs to usually a specific phenotype of interest to us or a specific disease, so that we can find DTU events that are potentially correlated with or causal to that specific disease. And because we're working with RNA-seq dataset, there's always going to be some level of noise. So this is kind of the more traditional framework that we've been working with in traditional DTU analysis. And so far, so good, because we already have a lot of popular DTU tools that work quite well with this kind of analysis. But this type of simple picture that we painted here is going to present us with two major problems. The first one is the small sample sizes. And traditionally, it used to be that even the richest data set of this type of analysis would have about 10 samples in each group. And the more RNA-seq data becomes accessible, the more we realize that this is not nearly enough to actually estimate the technical and biological error and noise in these groups. And when I say noise, I mean any signal that is not related to the phenotype that you're looking at. So that includes individual differences. And then that problem is kind of resolving itself as RNA-seq becomes more accessible and the publicly available data sets are growing exponentially. But we also design our tool, we need to design our tools to be able to exploit this growth and kind of more powerfully estimate the transcriptional noise in data sets and reduce false discovery rates. Because let's face it, our field is a little bit plagued with false positives. You take any two groups and do a differential analysis and you have hundreds of gene hits with differential expression or DTU in them. So that's what we don't want. And the second problem here, and potentially the more important one, is this hidden assumption in this picture that there is within group homogeneity in the traits that you're looking at. What that means is that when you're comparing this case cohort to the controls, you expect all the individuals to be affected in the same way. So to be different from the controls in the same way. And while that might be true for simple and Mendelian genetic disorders, where you have a single gene being the root cause of the phenotype you're looking at, it is rarely the case for more complex human traits and diseases that we're tackling today. So for example, we know a lot of the cancer types, diabetes, heart diseases, psychiatric diseases have some level of genetic components to them, and they are heritable to a degree, but we're still struggling a lot to compare get to the root of their etiology because they fall into this category of complex diseases where you have, instead of a single gene, a multitude of genes and genetic events that are causing these individuals to be affected. So this is more a realistic picture of what you're dealing with in DTU analysis and any differential analysis, actually. So what you want to account for is both the Mendelian traits, where you have a single gene affected, and in case of DTU, looking at this gene that we've been using in our example, the control population has the first isoform as their dominant, and the Mendelian group has the second isoform. So they have a switch to the second isoform that later establishes their phenotype. But then you have another group that has a more intricate heterogeneous structure where you have a multitude of genes that are affected, and each of the individuals might actually present with a different subset or combination of these events. And it could be that these different subsets or combinations all lead to the same phenotype, or it could be that they lead to different subcategories of the phenotype. So for example, if this was a schizophrenia group, each of these combinations, unique combinations, might actually be related to a different subtype of the disease, such as paranoid schizophrenia or catatonic schizophrenia, etc. So what you ideally want to do is to be able to distinguish these multitude of genes, to be able to also figure out which different combinations and subsets they come in, and also what subclusters in your case cohort they might be correlated with. And that's what we try to design SPIT in mind with, to first exploit the growth in RNA-seq dataset to reduce false discovery rates, and also take into account the heterogeneity present in most of the RNA-seq analyses that we're doing. So when you look at that first gene, in the simple Mendelian case, you have, again, if you have all of the individuals here are color-coded based on their dominant isoform. So in the simple Mendelian case, you have the shift from isoform 1 to isoform 2 in the entire case group. But then in the complex cohort, you have, instead of the entire or the majority of the case group being affected, you have only a subcluster of the individuals affected. And to complicate things even more, these individuals might not necessarily fall into the same subcluster, because if you look here, for example, the gray and orange individuals, they all have the first isoform affected, but because they have other combinations in the other 4 genes, they might fall into different categories. And if you look into the second gene and the third gene and so on, you see different subclusters going on with all transcripts. So what you want to do is to figure out which samples are affected for each of these genes and transcripts, and then to be able to figure out which samples cluster together and which subclusters are kind of closer together, and what is their relationship to the other subclusters present in your data set. So looking at the first isoform and going back to that definition of isoform fraction, what you expect in the control group is to have majorly really high isoform fractions for that first isoform, because it is the dominant one, so it accounts for most of the gene's expression. And if you look into the case group, you're going to have the implicated individuals kind of shift to the left of this distribution because they have the other isoform as their dominant. So you kind of face this bimodal distribution where the global minimum kind of helps you separate the individuals with this gene implicated and the individuals that are not. So what you can do is to fit a kernel density estimator to look for a global minimum, which then allows you to flag these samples as the affected samples and then compare them separately to the control group, instead of doing an entire group comparison and potentially missing this D2 event. And we use a rank sum test to compare all of these individuals. Because we do this type of test for all of the transcripts in our data set, there's always going to be a problem with multiple hypothesis testing. And you're going to face a decision between over-the-counter tools for multiple hypothesis testing correction, and they will work differently based on the level of noise present in your data set. So sometimes Benjamini-Hochberg is too permissive, but Bonferroni can be too stringent, etc. And unfortunately, you rarely know the level of noise present in your data set prior to doing your analysis. So instead of kind of putting your faith into an arbitrarily chosen correction tool, we ask ourselves this: because we have enough samples in our data set, what would happen if you were to take this group where you have no signal related to the phenotype because they're all controls, and if you were to randomly split it in half and compare them to each other, how low could your p-values get because of simply the noise and individual differences in your data set? And we do exactly that iteratively several hundred times to gather a null p-value distribution, which we then use to set a significance threshold and control false discovery rates. For performance testing, we did kind of recreate both scenarios of the Mendelian and the complex diseases. For the simpler case, we adopted the swimming downstream DTU simulation by Love et al. And they have more of a homogeneous structure, more traditional DTU simulation here with simulated reads. And for the complex case, we introduced DTU into 235 GTEx samples. They were originally all healthy samples. We put them arbitrarily in two groups, control and case, and then we introduced DTU cases in the cohort, kind of to recreate that heterogeneous structure that we expect to see in complex diseases. So we introduced 5 genotypes. I'll actually call them spliceotypes from now on. So we introduced 5 different spliceotypes, which account for different subclusters in the case cohort. And then we introduced DTU genes that can be shared and be affected by all of the subclusters, or could be specific to maybe 2 or 3, or could be specific to only a single subcluster. So it is kind of a more complex heterogeneous mosaic structure. And to complicate things even more again, we have different levels of noise in these two data sets because the first one is simulated reads and it has a much lower variance to mean expression ratio than the actual real reads coming from GTEx data set. Looking at the simple case, we saw that SPIT compares quite well with the traditional tools, but it gains significant sensitivity if you increase the sample size and kind of outperforms the rest of them. But more importantly, if you look at the complicated case, you see that the traditional tools mostly fail to hit a desirable false discovery rate because of the level of noise. And if they do, they kind of fail to get to good sensitivity rates to discover all of the DTU events that we introduced. But SPIT will consistently maintain a good FDR and TPR rate in this case. More importantly, because we introduced these spliceotypes, we later can kind of reconstruct them based on the specific DTU events each of these samples have upon that separation we did with the kernel density estimator. So upon finding all of these DTU events per sample, we are able to cluster them and then kind of also gather the information about how these clusters are related to each other based on their DTU landscape. And we did that 20 times and saw the same picture over and over again. Some interesting results that we did with applications of SPIT in real RNA-seq experiments is one is with schizophrenia. We looked at post-mortem brain samples from schizophrenic individuals and control adult samples. Specifically, we looked at their dorsolateral prefrontal cortex. And we not only discovered 6 genes with novel DTU events implicated in schizophrenia, but we were also able to find the specific subclusters of patients each of these genes are affecting. And some of them are shared among all of them, and some of them are unique to a specific subcluster. And all of these genes have been implicated with neuropsychiatric disorders and developmental disorders previously. So they do make sense a little bit. So another one, and this is the most exciting application of SPIT for me, this is when we had the SPIT applied to prenatal samples, that is before birth, prenatal brain samples, and regular adult and children samples, again, dorsolateral prefrontal cortex. And what we saw was that the transcriptional landscape in the developing brain is completely different than the adult or the children brain. So the brain has a different transcriptional preference when expressing the genes that it's expressing when it is in development compared to the adult or children brains. So looking at these implicated genes, and here the adult and children are the green ones, and the pink ones are the prenatal, so before birth samples. And in some of these implicated genes, you see kind of an entirely symmetrical shift of the preferred dominant isoform. And most of these genes have been implicated in autism models and have been proved to affect cognitive and brain development properties in mouse models. Okay. And this is just to show the coverage of this particular gene, that it's having kind of like a textbook example of D2U shift. And we have more of these genes, and I'm happy to talk to you about them if you have any questions about them after the talk. And recently, I've also kind of replicated this analysis in mouse brain when we saw the same thing, same phenomenon in the mouse brain, that the genes kind of shift to different isoforms when the brain is in development. And also, most of these genes are shared with the human experiments. So they are kind of orthologs. The majority of them are orthologs of the implicated genes in the human analysis. And further, because this data set that we adopted from Marcus et al. Provided us with 2 different neuron types, not only were we able to find the D2U events between the prenatal and the adult samples, but we were also able to cluster them based on the specific neuron type that each sample belongs to. And that is something that you would not be able to do with the traditional way of D2U analysis. So check out our paper and our Google Colab environment. It's an easy tutorial on how to use SPIT. And with that, I thank my wonderful advisors and lab mates, and I can take any questions.
Thanks. Very interesting talk. Thanks. I have a question for the post-mortem samples. Did you take into account the time from collection?
Yes.
And have you tried the method in cancer sample, which is like the paradigm of heterogeneity?
Could you repeat the second question?
Whether you tried in cancer sample like TCGA or...
Oh, not yet. We did not apply it on cancer samples yet. But to go to your first question, we did take into account the RNA integrity number, which is highly correlated with the time interval that passes after death and until you kind of collect the sample. And we did filter out all the DTU events that kind of correlate with RIN more significantly than the actual phenotype.
Thank you.
Yeah.
Very nice talk. Is SPIT agnostic to how the transcript usage is quantified? Like it can be used with short read data and long read data or single cell data or bulk data?
Yes, it is. So you apply SPIT after you do your RNA sequencing, and then you do your quantification with any type of quantification tool of your choice, so Salmon, Callisto, whatever, and then you take the quantification matrix of transcripts to samples just to count data, and that's what you run SPIT on. So in the Colab environment, there is specifics to different types of data sets. So if you have, for example, single cell, you might want to filter your data set differently, but it is agnostic to quantification in general.
Thank you.